Hi everyone, my name is Lynn Carpenter and I'm going to be your instructor this term, and I just wanted to welcome you quickly to Bio 171, and let's go ahead and get started. I can definitely tell you that this is one of my favorite classes to teach. Intro bio is always super fun for me, and ecology and evolution is a awesome area that I didn't even know existed when I was an undergrad. So when I was in school long, long ago, as my husband and daughter like to say, I this college I went to only went with zoology or botany. And so I didn't even know what ecology was, much less evolution. So once I discovered this, I thought it was the coolest thing ever. I'm an advisor in addition to being a lecturing faculty, which means I have the same degrees as everybody else, but I decided to focus on teaching and advising because that's what I love. I still do research, but it's on the side. Now, if we were still in person, I could tell you my office was in 3108 USB. However, it doesn't really matter because I can't get to my office because much like you all, I'm locked out of the building. It's kind of, I mentioned I'm going to be glad when all this is over. The one thing I can also tell you, though, is my research used to focus on ancient DNA as well as plant response to climate change, and I drove to Alaska and back for a field season, which was actually pretty cool. So I've been very lucky in my academic career and very thankful for the opportunities I had, and I definitely had a whole bunch of fun. So the goals of this class are to explain concepts in ecology, genetics, evolution, and again, that's my favorite part of science. I also want to teach you how to think like a biologist, okay? So very often, especially when I was an undergrad, you were just told certain things and you're supposed to believe them, but I want you guys to learn to be skeptical, especially in this day and age. We'll also learn to use biological principles when evaluating information, and I want you guys to be able to judge the media. It's so important for you in this day of all days to be able to, pick out the good information from the bad. And then also talk about other organizations, other organisms, with which we share this planet and why it's important to keep them around as much as possible. So I know you guys just got to the Canvas site the other day, and some of you are still working on getting the textbook, so don't panic if there's lots of time. However, there are activities for you to do before class starts, okay? And the research has shown, and you guys will learn soon, that one of my favorite things to do is to teach students how to study as well. In fact, I have a class on it that some of you might have taken or be interested in taking a different semester or so, but we're learning how to study. Okay, so you have to do some pre-class preparation that helps you learn. Also, I want you to focus on active learning. There's nothing worse than reading a textbook and then reading it 5 times again because you don't know what it said the first 4 times. Okay, that's boring and hideous. Also, we learn from each other. Okay, so we're going to have a lot of peer learning and discussions. Okay, we will have assessments, i.e. tests. The research has shown if we don't test you, you guys won't study and then you won't learn, and then this whole thing is pointless and that's no fun. And so, research supports the effectiveness of these methods. So we're not doing this just for fun. It's to truly help you learn the material. So the textbook we're using, and I'm not going to lie, I hate this book. It doesn't have enough information. The reading is scattered all over the place. Okay, it's not my favorite. If you plan on taking 172, though, you've got to get it, okay? And it's biology, how life works. There's even some mistakes in this book, and we'll talk about that when we get there, okay? But the other thing I want you to really pay close attention to are the slides and the lectures. Now, I will upload the slides every day, and you guys, my suggestion to you is to print them out, and then once we're actually going through lecture, to take notes on them, or at least have the file up at the same time that you do the lecture that's recorded, okay? And then you can take notes right on the side. And it goes best if you handwrite them. It's hard to remember them. However, my handwriting is atrocious, and so if you choose to type it, I understand, and you guys have to ultimately choose what works for you, and as long as it works, you guys do well. That's the most important thing. So, my few helpful tips and suggestions. Do not put off listening to lecture for more than a day, okay? Exams come straight from lectures, and I cannot emphasize this enough. You will be asked to use lecture material as your primary source. You can have other additional sources. In fact, it's encouraged to do so to help you put together big ideas on exams and so forth, but they must be as a supplement only. In other words, tests are open note, open book, but you can't Google your answers. You won't get credit, okay? So there's a lot of misinformation out there. I want you to use the lecture as the primary source, and then you can supplement it with additional source. You guys are welcome to share notes if you want. Some people in previous classes of mine have started Google Docs, and then you guys share notes back and forth, and that's fine. So you can totally share notes, and you're encouraged to do so, but you cannot share exams, okay? And we'll get to the format of exams soon, because I've told you they're open note, open book. but you cannot share the exams because it's a test. You guys have two amazing GSIs. They're awesome, Jake and Chloe. And so they will lead the discussion sections. Now, it's a little tricky because discussion sections are live, okay? And so you got to find a way to work through it with them if something goofy comes up and you can't attend your discussion section. But I want you guys there live because otherwise there's not much to discuss. Lectures, obviously this is recorded and you can listen to it at your leisure. Okay, I promise to try to get them up earlier than the day of lecture. I'll admit it, I was running a little late this morning. So, however, the discussion sections, they will be live. So the idea is that it's to reinforce lecture material. It's to also help you connect research to your life. It's to help you think like a scientist, improve your process skills. It's a great time to ask questions, review material, work on problem sets with peers, and get your pre-discussion work done before you show up, okay? Again, I want you to do well in this class. I know what's coming for you if you're going to take 172 or additional bio classes. Use this time to prepare, to learn how to study, and to learn how to do what you need to do to be successful. Okay, I just realized looking at this slide, and by the way, I have 4 dogs, and since I don't have an office anymore, I have a small animal rescue, and it gets a little loud in the background despite my best efforts. So if you hear barking, that's what's going on, and I apologize. We have got several exams in this course. They're actually worth 100 points apiece. Okay, so I just realized that this is wrong. They will be open note, open book. Your discussion and participation will be worth a significant percentage. Go check your syllabus out. I need to fix this slide and I will end up fixing it and then putting it back into the lecture later on today. So there is an extra credit paper. You guys get extra credit for also submitting the course evaluation. Did I mention extra credit? Okay, so my goal is that I try to give you guys a ton of opportunities to get extra credit. So if you want the grade, you know, a certain grade, you've got plenty of chances to get it, okay? But that does mean I'm pretty strict about grading in the end because I try to be very generous with the grades. And if you're a couple points away, well, and you didn't do the extra credit, sorry, that's kind of the way it works out. But I have complete confidence in you guys that you'll get the grades you want, and I want you all to do well in this class because that makes me happy. So the Science Learning Center is an amazing resource. I highly, highly recommend that you guys use it. It is now all virtual, of course. Sign up anyway. It's completely worth your time. They help you really learn to master the material. They have practice questions. They have all sorts of good stuff. Go ahead and sign up and take advantage of them. Definitely for this class, especially if you plan to take classes in the future. They're amazing, okay? So I can't say this enough. Bug them, use them. It's a resource I wish I'd had when I was your age. Any questions? Ha ha ha, I know, bad joke. I miss teaching you guys in person. However, my goal is to get this uploaded by 2. And then from 2 to 4, or let's say 2 to 3, I will be online. So as you guys are listening to this from 2 to 3, I'll be in the chat room. And so if you guys have questions, you can bump into the chat room on Canvas, and I will be there, let's say, from 2 to 3 today. Okay, and again, today's Wednesday. So should be plenty of time if you guys have any questions to let me know. The slides are formatting a bit wonky. I apologize about that. I use a particular program to record lectures so I can upload them to YouTube. And sometimes the slides look strange, so bear with me. I have to see if I can fix that. But to be honest, the program is throwing a fit this morning, which is why this lecture is a little late. We're going to go over course information today, which we've just done. Okay, now we're going to talk about characteristics of living critters. Okay, then we'll talk about DNA, which is basically gives us all of our charming characteristics. We'll talk about evolution in the tree of life, okay, and all sorts of other good stuff. In our Canvas webpage, go to Lecture 1 and make sure when you guys can and you get your textbooks that you can do the reading and answer the big picture questions, because a lot of the exam questions will come from that, okay, and obviously it's material that we're going to cover. So what I want us to consider right now is what it takes to separate living from non-living entities, okay? And I just realized I forgot to fix this slide for our online version. Talk to your neighbors, ha ha ha, okay. Did I mention I miss seeing you all in person? It's so much more fun that way. Anyway, I'm glad we're still rolling though, I guess. So think about it. What do you do to separate living versus non-living? What does it mean to be living? Okay, so for yourself, I want you to think of stop, pause lecture, hit pause, stop, whatever you want to do, and then take a moment and write down 3 things that you think it means to be alive. Okay, and then you're going to kick lecture in again. So as we're thinking about this, okay, and what it actually means to be alive versus not alive, think about those characteristics. And by the way, if you see a star in this lecture, that means write this down because you're not going to get it anyplace else, okay? In your slides, I left a blank. Write this down. And anybody who has ever had a class with me knows when I say to write something down, you should write it down, okay? So life is composed of one or more cells. Think about it. Everything we know as being alive is made up of cells. Predominantly made of carbon and water. Okay, that should kind of make sense. Acquires energy and material from its surroundings. Of course we do. We consume food all the time, photosynthesis, plants, whatever. Okay. Contains heritable genetic information. Now, genetic means it's coded into our DNA. Heritable means it gets passed on from one generation to the next. Can respond to environmental stimuli. Okay, it's cold today. I'm wearing pants, all right, not shorts because I'm responding to my environment. I'm layering up because I'm old and cold, okay? Can reproduce. That's something else, all right? Pass on those traits to the next generation. And then populations can evolve over many generations. Now, individuals do not evolve, okay? But populations do, and we'll talk more about that later in the semester. So write all this down. Again, you see the green star. You're only going to get it from lecture, which means write it down. Now, a really cool example of something that may not necessarily appear alive, but actually is, are stone plants, otherwise known as lithops. Now, I'm not sure if you guys ever seen these before. They sell them as novelties. I've owned many, and as my husband likes to say, I have the black thumb of death, so I've killed many, not intentionally. They are succulents, okay? They store a lot of water in their fleshy leaves, and the whole idea is they look like rocks, okay? So in each of these pictures, look at the upper right-hand corner. You can see how they're designed and to look like rocks because then they can avoid herbivores, okay? They will eventually, though, produce flowers after a rain, and they reproduce quickly. I can tell you, if you ever see one and you guys decide to purchase one, follow the watering instructions carefully, okay? They are not like other plants, and if you water them too much, they die fast, as I have found out the hard way, all right? But just looking at these guys, you may not necessarily realize that they're alive until they actually produce a flower. And then it's like, oh, okay, yeah, I can see how that looks alive and is alive. So here's a question for you, okay? And it's particularly pertinent right now, all right, with considering what's going on. Do you think viruses are alive, and why? Okay, so once again, pause the lecture right here, and I want you to either say yes or no. Go read the article that's listed there. It's actually pretty interesting, okay? And then once you've made your decision, you've read the article, then I want you to come back and we'll have a chat about whether or not viruses are alive. So let's take this back to what we just talked about when we were talking about our lithop plants. Okay, so remember, characteristics of living things. Are they made up of cells? Think about a virus, yes or no? Okay, is it primarily carbon and water based? Do viruses get energy from the environment? Do they have heritable genetic information? Okay, do they respond to environmental stimuli? Do they reproduce? And do they evolve? So stop again. Think through this and figure out whether or not you think viruses have these traits or not. So if we go through the list, okay, and I were to answer these, are they made up of cells? No, they're really not, okay? Remember, a cell, as the way we think about it, has a cell membrane and everything else, and viruses are a bit different. They're not made up of traditional cells the way we think. Are they carbon and water based? Yes, absolutely. Do they get energy from the environment? Well, yes and no. They get energy from their host, okay? They do have heritable genetic information. Do they respond to environmental stimuli? Well, they're inside the cells generally, so most of the time not. They do reproduce, though, and they definitely have populations that evolve. So viruses kind of straddle the line between yes and no. And honestly, scientists have put them into categories based on what part of their life cycle they're in. If they're dormant, they're not considered alive. If they're not dormant, then that particular time, they are considered alive. So it just kind of depends. And oh, look, a green star. Write this down. Think back to your high school days and think back to the whole idea of kingdom, phylum, class, order, family, genus, species. Do you guys remember that? Do you also remember a way to remember all of that? For me in school, long, long ago, as my daughter likes to say, in a far away and different time, it was King Philip came over from Greece Saturday. That's how you kind of remember all of that. And Carl Linnaeus is the one who came up with it. Well, back in the 70s, another scientist by the name of Carl Woese came along, and he did something incredibly creative. And you guys are going to somehow watch a video on this, but I haven't quite figured out how. I think I'm going to try and upload the video to YouTube. We'll see how this is going to work. I'm going to try and upload the video to YouTube, and once everything is up, I will then send instructions in the announcements for today, okay, on how to watch lecture and the videos and so forth. But anyway, Carl Woese, he was a very interesting guy. He took a gene that was conserved, meaning every single species on the planet, every critter had a copy of this gene. And he was using genetic work to try and understand how all of these critters are related to one another, okay? And the way we treat this now is that he found a new way to group things called domains. There are 3 domains. There are eukaryotes, which is us. Think of what it means to be a eukaryote. Hint, if you don't know, go look it up, okay? The bacteria, the guys that, we interact with on a regular basis that we know and love today, as well as archaea, which is kind of the old bacteria, okay? And it was Carl Woese that came up with this. And in a couple of slides, I'm going to ask you to go watch the video, which I'm hoping to be able to successfully upload to YouTube. If not, we'll come back and figure it out. So, but that kind of sits above kingdom. That's the way we treat this. So there's domain, then there's kingdom, phylum, class, order, that type of thing. That's how we work today. Oh, look, a green star. You should be writing this down, okay? So the eukaryotes, remember, eu means true, karyote means nucleus. So they have a nucleus, and by the way, that's us, okay? What's interesting about this tree is that the archaea are more closely related to the eukaryotes. From a scientific perspective, that makes no sense, okay? The group that used to be considered prokaryotes, just because they don't have a nucleus, those were the bacteria and the archaea that were grouped together at one point in time, but now they're separated because the bacteria, genetically speaking, are distinct from the archaea, which are considered to be the old bacteria that evolved first. And again, there's a lot of questions on this with regards to science and how things are really related, but I just kind of want you to know how this is organized and, how the classification system works. And, science is always changing. That's the cool part about it. And so because science is always changing, then we learn as we go and we modify, and someday things might be, hopefully they'll be worked out and we'll know a lot more, of course. So we do not give the archaea and bacteria anywhere near as much credit as we should. Okay, so these guys live everywhere. They live in environments that would kill us within seconds, okay? And they somehow manage to thrive in these environments. If they live in extreme conditions, they're called extremophiles, okay? They can be at the bottom of the ocean in hot thermal vents, under enormous amounts of pressure. They can live on glaciers. I thought I was whining about being cold last winter, okay? So the archaea live in extremely hot, sulfur-rich water. They can be in the deepest part of the oceans and the uppermost parts of the planet. They are everywhere, okay, which are actually incredibly cool. And whether we realize it or not, the bacteria and archaea were here way before mammals and our brethren ever were. Okay, so our ancestors, Homo sapiens, we were lucky to be here about 200,000 years, if that. The bacteria and archaea have been around for billions of years. They are awesome and fascinating, and we're only starting to learn about them now, and we will definitely revisit them, more information about them later on in the term. The other things that we may not realize with the bacteria and archaea is they play critical roles in nature. They give us oxygen in the atmosphere. We tend to think of trees as giving us oxygen in the atmosphere, but the reality is it's the bacteria, the photosynthetic bacteria and so forth, the tiny guys that actually create the most. So the next time you guys wander by that pond scum and you're all grossed out, thank the pond scum, because that's actually what gives us most of the oxygen in the atmosphere. They fix nitrogen for plants. This is what allows us to live. They help things to decompose, and we might take that for granted, but think of how gross life would be if nothing ever decomposed. We'd be over our heads in dead things. That would be horrible. Okay, they help nutrients cycle. They are important for mutualisms. You guys have bacteria in your gut. You've heard of all the, Activia commercials and so forth, and they actually help us to digest our food. And bacteria are much better at digesting our food than we are. okay? And we'll come back and talk about it later on in the term. But bacteria actually produce, on average, over 300 enzymes to digest our food, and our gut only produces somewhere between 70 and 80. So that tells you they're way better at food digestion than we are, okay? So without them, we'd be pretty, pretty much in trouble. So thank your bacteria, even though we can't see them. Okay, so as we are chugging right along, we're going to start talking about the DNA. And I'm sure you guys remember DNA. That's all those A's, T's, G's, and C's that give us our charming characteristics, okay? So this is what encodes our information to give us our hair color, eye color, height, all sorts of other good stuff. We'll talk about it now. So let's talk about that genetic material. Okay, so we know that the structure is a double helix, and I don't know if you guys know the story behind how the structure was discovered. It was discovered in the 1950s by two scientists named Watson and Crick, and it turns out they were kind of jerks, believe it or not. I always swear I'm going to create a class called The Scandals of Science, because it's fascinating to me how scientists can sometimes not always make the best decisions. Okay, so the way the story goes is that Watson and Crick, no, you don't have to know this for the exam. Yes, you have to know this in general because it's good for you to realize where science came from. Okay, Watson and Crick were working on the genetic structure of DNA, and they couldn't figure it out for the life of them. At one point, they thought it had triple helices. At another point, they thought it had a single one. They weren't really sure how to put it together. Down the hall from them, there was a scientist by the name of Rosalind Franklin. And apparently she was a tough lady. And to be honest, you kind of had to be a scientist back in those days, because women were, realistically speaking, were not treated very well. Thankfully, it's different today. Okay. However, she was tough, and she had to be. She worked on X-ray crystallography of DNA, and she wasn't necessarily trying to research the same thing as Watson and Crick. But unfortunately for her, one of her graduate students discovered that one of her images really would help out Watson and Crick as they're trying to figure out the structure of DNA. So her graduate student, and this is highly unethical, by the way, her grad student took that picture of the X-ray crystallography of the DNA, ran it down to the lab down the hall, showed Watson and Crick, who then realized it was a double helix with the bases in between, ran it back to her lab, and then put it where it belonged, and she never knew what happened. So Watson and Crick get all of the, glory and accolades for discovering the structure of DNA, where the reality was they wouldn't have done it without Rosalind. Now, the really part that I hate is the fact that they never thanked her. They never told her anything good. They never acknowledged her contributions until the book, The Double Helix, was written. Okay, in which case that book was written, and that's when they finally admitted to it. All right, but that's unethical. We don't do that. And the even worse part is they won all these prizes and accolades for discovering the structure of DNA, and she never even knew her contribution because she died, and they would not give the award posthumously, which means once you die, you can't get the award. Okay, so she never got the recognition she deserved, except those of us that know the story try to spread it around because she deserves accolades and recognition for it. The other thing is, if you guys like to read about science history the way I do, yes, I'm a geek, I know. However, there's this really cool book called The Dark Lady of DNA about Rosalind, and it's interesting. It's an easy read. It's written for everyday people, and it kind of goes through and tells the story to explain what happens. If you guys are interested, I would highly suggest reading it. But anyway, that's the story behind how the structure of DNA was found, and another reason that I want to design a class on the scandals of science. So with regards to the diversity of structure of DNA, what this means is you can have different combinations of nucleotides and create different DNA molecules. So what I want you to do now is stop the lecture, look at the slide, and then what I want you to do is to write down who pairs with who, okay? So use your knowledge of DNA to fill in the right side of this double helix, all right? And that way, if you happen to ever see this again, you never know, you'll know how to fill it out. So take a couple minutes and do this now. Interactive lectures are important, otherwise you guys would fall asleep on me. So on the bright side, the fun part of giving lecture at home is that I've got the most awesome husband who gets interested in things that I say during lecture, and then he looks up information for me. Apparently, Rosalind had an entire university named after her, yay, in Chicago, so that's pretty cool. So she did get some recognition. Now, I don't know why the slide looks the way it does. Something about transferring it over. You guys can still get the information you need out of it. Just ignore the white boxes. Oh, look, a green star. Write this down. okay? Those are the base pairs that you should have been able to write down, as pairs. So remember, A always pairs with T, G always pairs with C, okay? Super easy peasy, no stress. Then you just go through and write everything down. So, having the ability to replicate is incredibly important for DNA, and this will turn one double helix into two. Now, we are going to go in depth into how this process happens later on. But what I want you to remember just for right now, okay, is you have one double helix, it splits apart, and then the other, base pairs will line up with who they're supposed to match with, and you'll end up with two double helices. That's the whole idea. Okay, so as this process happens, oh look, a green star, write this down, okay? The two strands of DNA are separated, and this happens by an enzyme, by the way, if you happen to be curious. The idea is that each strand is used as a template for the new strand. So you have one old strand and one new strand, and this is called semi-conservative replication. Write that down. Semi-conservative replication, meaning you got one old strand and one new strand, and we'll go into more detail later on in the term. Okay. However, this is important to know for now. Now, there we go. The slide is conveyed properly. So, having a few technical difficulties, bear with me. I'm adding new things into this lecture, and so somehow there's technical difficulties. Mutability, this means you can add mutations. This is why that we all are different, okay? And so if you get errors during DNA replication, you introduce mutations, and this creates genetic variation. Now, from an evolutionary perspective, variation is a good thing. You need to know this, okay? The reality is that mutations happen randomly, okay? But those who have the better mutations will do better, and they will be more successful and produce more offspring and all sorts of other good stuff. At least this is the way it's thought of now, all right? So mutations generally happen randomly. If somehow you have, a C gets changed into a T, from that point it gets carried on and everything changes. So it's interesting. All right, now what I want you to do is to take a moment and pause lecture, okay? And I want you to consider, what are proteins and what do proteins do? We will go over in the next slide, but I want you to take a few moments, fill this out, and write down what you think, okay? So, well, the reality is you guys can pause this lecture at any point in time, get up, stretch around, but after a while, you're going to need a break. So try and turn this, even though you guys are listening to a recording, turn this into as active of a exercise as you possibly can, because you're much more likely to remember it and you will do better overall. So let's talk about some examples of proteins. Hemoglobin is a protein. What does hemoglobin do? Remember, hemoglobin is what binds to oxygen, which is what allows our blood cells to carry oxygen around. Incredibly important, okay? Insulin regulates our blood sugar. Lactase help us to digest milk, okay? Collagen, a structural protein. As you get older, the collagen breaks down and everything goes, but we'll talk about that later, okay? Myosin is a motor protein. That's what allows our muscles to move. So proteins basically help us do everything. They help us function. Without them, we'd be in big trouble, okay? And what codes for those proteins is that DNA. And so that's why DNA is so incredibly important and gives us these traits. Now, if you guys ever hear the term central dogma in DNA, this is exactly what it means. Hint, you should know this, okay? So the first thing we should consider is that DNA is what stores the information. DNA goes through transcription to make messenger RNA. Messenger RNA carries information. That is then goes through translation to make proteins, okay? And so this is how the central dogma actually works. Now you might be asking yourself, what truly is a gene? Well, a gene is a section of DNA that contains information for building a particular protein, okay? So if I ever ask you to define DNA, that's what it is. Or define a gene, that's what you should know. Oh, look, another green star. You should be writing this down. So the process of transcription is regulated by what people or scientists think of as non-coding DNA. And initially, scientists used to think that that was just junk DNA. Well, it turns out that the junk DNA might not be as much junk as we thought it was before. In fact, this might actually control what genes are expressed and when they're expressed, meaning turned on and off, and when proteins are made and so forth. So it actually turns out that might be incredibly important. And then the translation is using that genetic code to create things. Okay, so just to give you an example though, messenger RNA carries the information, but also realize that there's other types of RNA that are used, the ribosomal RNA, which provides structure, and transfer RNA, which carries amino acids in and out as proteins are being synthesized. This will make more sense in the next couple of slides. So right now I'm just kind of giving you guys these terms, and then we're going to actually apply them in the next few slides. So if they don't sound familiar, they will in a few minutes. So transcription is the process where DNA is made into RNA, okay? And so it's messenger RNA. During this process, and the way this is made, of course, is once again complementary base pairs. So the thing to consider, though, is that in RNA, you don't have thymine or the T, you've got uracil or the U. So A pairs with U and G pairs with C. Okay, and so as the RNA is made, that's actually how it's made. Once again, it's all based on the DNA, and that's what gives it its characteristics. So transcription is the process used to make that messenger RNA, okay? And then that messenger RNA is used for translation to make the proteins. And so once again, you have complementary base pairs, okay? And the whole idea is you have triplet codons that the transfer RNA will bring in amino acids based on what the codons are. Okay, and so I'm going to, in today's announcement, I will also add some interesting videos that kind of help explain this, because a lot of times, you know, picture's worth 1,000 words. Think back to high school, all the topics you tried to forget, okay, and realize that those amino acids are brought in based on the triplet code, okay, and so each amino acid has one particular code, and that's how it's paired together. And then those amino acids are brought together kind of like pearls on a necklace, and then that's what constitutes the primary structure of protein. Okay, and so later on we're going to talk about primary, secondary, tertiary structure of protein, but what I want you to know is that the protein is made up of the amino acids. The order of the amino acids is determined by the messenger RNA, which was, the order of that was determined by the DNA as it went through the process of transcription. Okay, folded proteins then act as enzymes, as structural proteins, as membranes, hormones, signaling, all sorts of other good stuff. Proteins are incredibly important for us and our bodies, and basically it's how we function. Oh, look, another green star. You should be writing this down. The messenger RNA, which was created through what process? You guys should know this. It starts with a T. Okay, messenger RNA, which was created through transcription, okay, is then used in the ribosomes where the transfer RNA brings in the amino acids. Which amino acid is brought in is based on the triplet codon. Those amino acids are put together. It's the primary structure of the protein, like pearls on a necklace. That protein then will fold and is used to make, you know, all sorts of things in our body to function and so forth. So again, I'm going to show some videos, some additional videos in the announcements in class, and I want you guys to watch them because it does definitely help with understanding how this process works, and you do need to understand this process, okay? So I want you to understand the central dogma, the process of how proteins are made, and all sorts of other good stuff. What I would like you to do now is to take this slide, okay, and I want you to write out the function of these different types of RNA. You need to know this, okay? So go write out the function of these different types of RNA, and then take a few minutes to do that, pause the lecture, and then when you're ready, go on to the next slide. So let's talk about this genetic code. Okay, so there are 64 different codons. Each of them are 3 base pairs long. They all code for only 20 amino acids plus 3 stop codons. What does this mean? This is called redundancy. Okay, and so more than one triplet codon will code for the same amino acid. From an evolutionary perspective, that's a good thing because that means you can have a mutation and it doesn't necessarily cause any issues for the critter. Okay? The second thing, do not memorize this table. Can you imagine how awful that would be? Okay, but you are expected to be able to read it. So let's talk about how to read it. The first of the 3 triplet codons is on the very left side. So there's a U, a C, an A, and a G. For example, the U's are all the way across the top. The next 4 would be the C's all the way across, and then the A's, and you guys should be able to see how to read that first base. The second base, okay, look at the blue, and it says second base across the top. Now, for example, the second base is the U, that in that row, that second base are all U's. In that next row, they're all C's, okay? So, for example, you know, we've got a C for the first base, all right, and then a U Okay, and you can guys see how it should be able to see how to read the table. Now the third base pair is the green and it's all the way to the right. Okay, so for example, if you go all the way to the top right, okay, you will have a U, a G, and another U. Okay, and then that tells you what that particular code, which amino acid it codes for. You should be able to read this table. You're going to see it again. If I mentioned you're going to see it again, because you're going to see it again. Know this. So here's an application question for you. Okay, and which nucleic acid is represented in this code table? Now, based on 2 slides ago, you should be able to answer this. And it's not C. Okay, so stop for a moment, think about it, and then come up with the answer to this. Hint, hint. Look at that first base and that second base, and look at what the code says. Okay, do you guys know the answer yet? It's RNA, because remember, RNA has uracil, not thymine. In addition to that, this genetic table codes for messenger RNA, so we kind of know that anyway. Do you guys see how this works? I want you to not just learn this information, but be able to apply it, because you've got to be able to do this for 171, because 172 is going to be just like it. And that's kind of what it's like taking science classes here. All right? It's my goal to get you guys ready, and that's a goal I take very seriously. So now let's go to that evolution and tree of life. Okay? And so now we're moving on to the last part of lecture. And let's talk about how life evolved and all sorts of other good stuff, and also how to read trees and more interesting things. So what I want you to know is that all living things share a common ancestor, okay? We evolve through descent with modification. This is what gives us the diversity of life. And this only happens when you have traits within a population that are variable. That variation is heritable, meaning you pass on from one generation to the next. And some individuals reproduce and are more successful and have more children than others. If that's the case, then your population will evolve. Make sure that you can write this down, okay? One of the best ways, by the way, to study, and again, I teach a class on learning how to study, so you guys are going to get stuck listening to me giving you hints, because I want you to ace this course and do really well here, okay, is to take out a blank piece of paper and write everything you know about that concept, okay? So what I would do is I would study this, okay, and then I would pull out a blank piece of paper and then tell me everything you know about evolution, and you should be able to list these 3 things for sure, okay? So, and then also you should be able to go through the next slide, which is an example of evolution through natural selection. So let's talk about that now. So how evolution happens is you have a population with variation. Those traits get inherited, okay? Certain individuals with certain traits will get eliminated. For example, the bugs that are easier to see get eaten by the birds. And then those that survive will reproduce, okay? And then there will be more of them because those traits that enhance their survival will make them have more children and be more successful, okay? Now, what I want you guys to be able to do, hint, hint, do this now, is to be able to come up with your own example of natural selection and evolution. And so what would happen if we were sitting in class is I would have you talk to your neighbors and say, okay, come up with your own example of how a population would evolve. This can be a fictitious one. This can be fake. You can talk about dragons with purple polka dots and how they might evolve. You can talk about insects. I don't care, but be able to come up with your own example of evolution through natural selection. Okay, so take some time to do this because it's really important. I want you to be able to apply this concept and not just memorize it. Now, this relationship that is shown between individuals as they evolve and diverge from a common ancestor is generally represented with what are called phylogenetic trees. Now, this particular tree and diagram that I'm showing here, by the way, is a historical one, so there's a reason for it. It was one of the only figures that Darwin actually drew in his book, Origin of Species, okay? So this was a hypothetical tree that he came up to show how different individuals be related to one another based on a common ancestor. You're not expected to necessarily be able to repeat this, but just realize that, it's an important figure and it's one that was, is considered a classic in this day and age. So the way you actually read these trees are that the 2 species that are closest to one another are more closely related to one another. The further apart they are, the further, the relationship is. So given this information and based on the tree above, be able to answer which of the following is true. So what I want you to do is take a couple minutes, read through each of these possibilities, and figure out which one you think is true. So stop lecture here. Okay, and then go ahead and take a couple minutes to answer this. So if you guys have worked through this, you should find that B is true. Okay, so the reality is we can eliminate A because moss and pine are sister taxa, so you could either have it look like this or you could flip moss and pine and it would still be the same. Okay, so green algae more closely related to pine than moss. That's not true. Okay, once again, you can flip that. Green algae are related to red algae, but not related to moss. Once again, also not true. Okay, so when in doubt, use the process of elimination, figure out which ones you know are not true, and then go back and look at who's ever left and figure out, in order to circle it, as long as there's only one answer and you're told there's only one answer, you go after the only one that could possibly be true. By the way, this once again is an application question, okay? And so I want you to be able to apply the concepts that we talked about. So when you're considering your tree, okay, and you're learning how to read them, okay, you're going to be asked certain questions like this. Now, these questions are not meant to trick you, but they're meant to ask you to apply things, okay? And so what I want you to realize is by looking at this tree, you actually could kind of flip things around and have it still mean the same thing. And so the tree is going to look like this for this question. And the next slide, I'm going to flip the tree around, but I want you to realize it's the exact same tree. Okay, so this is not my favorite example, but we tend to share examples because I want to give you guys the same information as other students who take it the semesters that they take this course. And so take a moment, look at this, figure out which one shares a common ancestor with frogs. Now, take a second and do this, and then we're going to come back, okay? By the way, the answer is D. Again, this question's not my favorite. The next one's not quite as bad. So let's talk about this one, okay? So, by the way, exact same tree. But what I want you to do is think about what's different, okay? The common ancestors, what branches off of the common ancestors can be rotated a bit, and the tree means the same thing. That's what I want you to get out of this. According to this tree, which animal is most closely related to the lizards? Find your lizard. Find the two that are the most closely related. The answer is D. Humans and mice are equally related to lizards, okay? So then what I want you to do is go to the next slide because it's going to show you the two trees that are next to one another, how it technically means the same thing even though it's got this rotation. Now I know this seems kind of silly, but when you're tackling systematics and you're learning a new field, you got to learn all this stuff, the ins and outs, because I want you guys to feel comfortable reading a tree because that's incredibly important. You got to be able to know how to read a tree in this class. So let's go to the next slide and I can show you the trees side by side and you'll see that they're the exact same thing. So as you can see here, once again, the tree is the same, okay? But it's just the nodes are rotated. So take a moment, pause things here, make sure you can trace the relationships, all right? Because you've got to know how to interpret and read trees. Have I hinted on this enough that you guys need to know how to interpret trees and how to read them and figure out who's related to who? Okay, remember that your critters can, as long as they're on a node, they can spin and it means the same thing. Okay, so in other words, mice and humans are equally related to lizards. All right, and you can see it there. So it's not that, you know, take the tree on the right. You look at the human and you look at the lizard, but remember it can spin. Okay, so then look at the tree on the left. That's the same thing. Okay, all you're supposed to do is to be able to read it based on the information that you're provided. All right, so I just want you to feel comfortable with trees and feel comfortable reading them and, you know, realize that you can spin those around and it still means the same thing. Okay, so now, why might this be important, you might be asking yourself. And I would say, aha, that is an excellent question. Because scientists are actually trying to piece together what's called the tree of life, meaning all of the different critters and all the different species that we have on this earth. They're trying to use sequence data, morphology data, all sorts of other good stuff to figure out who's related to who and put together a big, huge tree of life. Can you imagine how arduous this process is? Okay, so it's incredibly labor intensive, but it's incredibly cool to figure out who's actually who. All right, now what I want you guys to do is to pull up your slide, the actual like PowerPoint slide, and zoom in, because I realize it's hard to see this, you know, based on the fact it's so small, but take the moment to see who's actually located where. Where are humans? Where are plants? Where's the bacteria? Where's the archaea? You're not expected to memorize it, but again, it's interesting and, you know, it's pretty cool to look at. So if all of the slides have worked the way I was hoping they would, as far as me inserting videos, you guys should have been able to watch the video on Carl Woese. Now, I will tell you, I don't really like the video that much because it kind of makes him seem like a conceited jerk. And I came from the University of Illinois and I met Carl Woese. I was very, very lucky. And the one thing I will tell you is that, the video doesn't really do him justice because really he was one of the kindest men I've ever met. And a friend of mine took a class with him. To be honest, I was too intimidated to take a class with him. But Carl Woese was the type of person that had reached the status at the university, and I think he's passed on since then, so I think he's gone now, where he could do whatever he wanted to with regards to teaching because he won so many awards for his work that he was kind of set. By the way, the rest of us will never, I will never achieve that status. Unfortunately. However, he did. My friend that took a class with him, one thing I'll tell you about him is that basically he wandered in and Carl would basically lecture on whatever he felt like talking about that day. And the students were just glad to be taking class with him. Again, I will never reach that status, okay? But the other thing I want you to get is that he was a very kind man. And he, there are a lot of scientists, even all the places I've been that are not always the nicest. And, aren't always the nicest to me either because I'm just a lecturer. However, he was one of the nicest people when I was a student, and that was something I always greatly appreciated. So the video, does a nice job portraying him, but they kind of, it doesn't show his kindness, I guess, is my frustration. So the tree of life that he came up with was based on ribosomal genes. And what I want you to get from this, first off, you've seen this again, right? Okay, we showed you this earlier. You watched the video, you're seeing the tree again. Why do you think RNA would be a good choice to try to piece together the tree of life? Think about that for a moment, okay? Why would RNA be a good choice to piece together the tree of life? I will tell you in the next slide. Oh, look, a green star. Write this down, okay? The reason that RNA is a good choice for the tree of life is every living critter has this gene. They have to, because RNA is one of the bases for life. Okay, and so that's why he was able to use this particular gene and to put together the relationships of these creatures to one another, which is actually pretty cool. All right, so today's lecture is a little bit short. Don't get used to it. I'm just trying to work through some of the technology, make sure the videos are inserted. However, you guys, as you all know, you're paying a fortune for this education. So I am going to make sure that you get your money's worth, okay? However, I want you guys to relax, to settle into the class, to listen to lecture, to realize I'm here if you have questions. I will be in the chat room from 3 to 4 today. 2 to 3? 2 to 3. 2 to 3 today. I will be in the chat room if you guys have questions, and hopefully this will upload in time. And then you guys do have discussion tomorrow, okay, where your awesome GSIs will teach you how to read a scientific paper, one of the most important things you will learn in science, all right? I wish you all a wonderful day. Stay safe and stay healthy, and I will talk to you very soon.